The evolution of DNA morphology and its coding regions has lineage to gene expression divergence in respectable species. For years, I've aggregated the complex nature of DNA and its analogous effect on the phenotype of an individual to the way genes express themselves. Resultantly, anatomical homology is not present within a group of species. My study of the article "Gene expression is coupled to evolution of DNA structure and coding regions" has further unveiled that phenotypic variation is linked to the divergence in gene expression of a specific gene. The aim of this research article was to explore various processes that cause gene expression divergence. How I understood this erratic exploration was through three processes. The first hypothesis is correlated with the proliferated changes in regulatory encoding sequences of the gene itself, CIS, and changes in the sequence of regulatory factors, trans. As seen, there is a specific coding region of the DNA strand which undergoes transcription and translation by the transregulators to create DNA-binding proteins. Genes are segments of DNA, and so gene expression is contingent to specific CIS regulatory regions. Gene expression divergence thereby comes from a modification in transcription regulators, as the binding of DNA and the activation of transcription in CIS is alternated to new instructions disparate to its pre-prescribed role. The second hypothesis is coupled to sequence mutations and transcription factors. An alteration in the fabrication of TFS inclines disruption in the process of transcription and its lineage to the production of proteins. TFFs are predominantly protein-formed. An example is activator proteins. These are aggregated complexes that allow interactions for diversified control of CIS. TFS control over CIS is facilitated through its recognition of nucleotide sequence motifs found before the gene in a region called the promoter, or after the gene in an area called the enhancer. Conclusively, the activator proteins adhere to the promoter region, forming a complex that generates binding and thus the initiation of transcription by RNA polymerase. A mutation in the TF aspires polarity in gene expression within a group of similar species. The final hypothesis corresponds to nucleosome positioning. Such positioning is determined by CIS effects. As CIS regulators aid in the coding of genes, they produce localities for nucleosomes, of which then facilitate the access of transcription factors to a particular motif, either promoter or enhancer. The concluding and scientifically supported outlook of gene expression divergence between species, explicitly the two disparate species studied in this experiment, Drosophila and yeast, is through DNA structure. CIS, as you will soon see, CIS and DNA structure share affinities. How? Well, in the experiment, 35 types of dinucleotide DNA structural scales were used as a backbone from which information on the structural properties imperative for transcription could be synthesized. Accordingly, the structure of DNA is important for protein DNA recognition. The specificity of protein DNA interactions resultantly initiate transcription, showing correlation with gene expression, as you can see. Further, DNA curvature corresponds to dinucleotides, as dinucleotides bend easier to facilitate the packaging of DNA into the nucleosomes. Thus, as nucleosome occupancy correlates with transcription levels, DNA sequencing is a critical determinant of nucleosome position, which, as we know, directs TFs to motors for gene expression.